Everyone, thanks for coming to the next rendition of Active Matters USA seminar. Today, we're delighted to have Katherine Copenhagen talk to us about topological defects drive layer formation and gliding bacteria colonies. Thank you. Oh, and I guess before we as usual, so if there's any questions from either the in-person audience or virtual, just please feel free to either ask it, or in the Zoom, you could also just type it if you rather not do it, and also try to moderate the chat. So let's get started. Thank you.
Cool, great. Thanks, Farzan, and thanks everyone for coming to watch this talk and learn about some topological defects in bacteria colonies. So this work is what I've been doing in the Shavitz lab here at Princeton. I'm a postdoc here, and other people have contributed are Ricardo Alert, Matt Black, Edwin Green, and of course Josh. So before I start talking about active matter, I wanted to just do a brief reminder of like passive materials and how we are kind of inspired to study these active materials. So in passive materials, you can use statistical mechanics to basically look at your system of molecules. If you have a bunch of molecules and you know sort of some properties about the individual molecules, like how fast they're moving, which could have to do with their temperature and their density, maybe if they have a magnetic moment or a weird shape. You can do a bunch of averaging over your different possible states that your molecules could be in your system. You can apply your laws of thermodynamics since we're in equilibrium, and then you can learn all sorts of useful properties about your macroscopic materials, like whether you'll have a solid, a liquid, or a gas, whether it'll be magnetized, how easy it is to break, all sorts of things that you would want to know if you're going to be building something out of a material, right? So that's kind of the direction that we want to go with looking at active materials. So now we have molecules, and this was kind of introduced by Vicek in 1995, where he took a system of a bunch of molecules, but now he said, okay, we're going to think of these molecules as being alive and propelling themselves in some direction. So now we have an active material and we're out of equilibrium. So now if we know something about our properties of our molecules in our system, like their speed, or somehow they're applying forces on their environment around them, which could be a propulsion or some sort of shape change, then you might also want to know about their density, if they have a magnetic moment or weird shapes, similar kinds of properties. And then if there's anything else you might want to know about an active molecule, then maybe we can do a similar process where we can apply a bunch of averaging and apply some laws. Now we can't use thermodynamics because we're out of equilibrium, but there's some like sort of laws or equations governing the materials that you can use. And there's a lot of sort of theoretical research around figuring out what these laws are, what kind of molecule behaviors can lead to different interesting materials. And then what kind of material properties are also interesting in an active material. So these exist all over the place in biology. So they are very relevant to the world around us. We have like bird flocks, fish schools, crowds of people, herds of wildebeest, all the way down to small scales like bacteria, you can see here. And then even inside of an individual cell, the cytoskeleton kind of makes an active material as well. So
Katie, what can you say? What is the cytoskeleton in the lower right? What type of cell is it? What are we seeing there?
So both of these are Myxococcus xanthus, which is the bacteria I'm going to talk about for the rest of the talk. So this bacteria is very useful and interesting to us, especially because the same bacteria just changes a little bit about how it moves by itself, and it goes from forming this kind of flat spread out thing to like this other phase, which is like aggregates or droplets, where it's like dewanted from the surface.
All right, thank you.
So yeah, good question. But yeah, so that leads to the next slide, which is what is this bacteria? So this is Myxococcus xanthus. It's a bacteria that's found in the soil all around us. It's totally harmless to humans. It survives by eating other bacteria. By itself, it's about 7 microns long and half a micron wide. So it's a long, skinny rod and glides along surfaces at about a body length a minute. We think this is a really interesting experimental system to study active materials because, yeah, you get a bunch of different phases and like macroscopic materials properties out of like the same bacteria. And since it's a bacteria, you can also manipulate its motion like genetically. change something about how it moves or interacts with other cells. So the developmental cycle of this bacteria, when it's in nature, involves this starvation response, which helps it survive when it runs out of food. So when there's a lot of food, when there's a lot of food around, it spreads out into a thin layer covering the surface that it's on. And this layer is about 1 to 2 layers of cells thick usually. And then as it runs out of food, it starts this process where it starts to form more and more layers on top of itself. And then eventually it kind of smooths out into these round mounds called fruiting bodies. And these things inside of these, the cells sporulate, and the spores can last basically forever until there's more nutrients available, and then they germinate and go back to this cell layer spread out. So we have a video, hopefully the videos play okay, but this video is taken by Cassidy in our lab, and you can see this layering process. So in this video, the dark patches are empty gel showing through a layer of cells. This is a first layer of cells, and then we have a second layer. A third layer has started to form here, and then a fourth layer, and then it kind of starts to smooth out and like de-wet into these fruiting bodies. And there you have that nice big aggregate of cells.
And are there cell divisions on this time scale of this nice movie, Katie?
Not in these conditions. If there were nutrients around, yes, there would be cell divisions, but since this is starvation media, so the gel, they have no food to consume and divide, so there's there's very few divisions probably in this video.
So
but they're done. Huh?
Great. So if you were to have nutrients and 2 of these colonies, because of the divisions, collided, presumably that would be another mechanism for stepping into the third dimension.
Yeah, so most of the activity, like the active material stuff that we see in this bacteria, or that we've studied or looked at so far, is driven by motility. So I am actually, the rest of the experiments I'm going to show are done at high nutrients, but that's only over the course of about an hour. The division time of the cell is about 6 hours.
I see. Okay.
So they move a lot faster than they divide when they have high nutrients. So I think most of the like active material like properties that they show are due to motility more than division.
Okay. And is it twitching motility? How do they actually move?
So there's 2 motility systems in here. They do have twitching motility with the pili, but most of the motility that they actually show seems to be due to this gliding motility, where they form a focal adhesion at the front of the cell. So they basically like attach to the surface, and then they have like a track inside of the cell body, and they pull that adhesion back along their track. And since it's attached to the surface, that makes the cell body like glide forward. And then it detaches at the back and reattaches at the front and like pulls itself forward like that.
So it's a little bit like a tank tread?
Yeah, a little bit, yeah.
Okay. Uh-huh. And then you mentioned they feed on other bacteria. So they're about the same size as their potential food. So do they explode it first and then take up the resulting nutrients? Or how
Good question. So that's kind of why it has evolved these like collective strategies to stay as a group of cells. So they hunt and like kill other bacteria by lysing them, but they have to be in groups to do this. So you need a bunch of myxo cells around, and then they eat like E. coli, for example. They'll like rub up against the E. coli, and then they'll eventually lyse it and then absorb the nutrients.
And do they lyse it by secreting an enzyme that attacks the peptidoglycan cell wall of the E. coli, or how does that? How do they actually get the lysing going?
Yeah, I'm pretty sure they excrete enzymes, which they have to like accumulate enough of too, which is why they need to be in packs.
Okay.
So individually, they can't secrete enough to lyse them out with another bacteria, but.
And have they been seen, have these packs been seen chasing E. coli? Can they detect them and sense them chemically and then have some kind of chemotaxic attack strategy?
There hasn't been any chemotactic like strategies seen in Myxo.
Okay.
So it seems like everything they do is from very local like interactions. So yeah, there are nice videos. I didn't put one in here, but of like a Myxo colony and an E. coli colony next to each other, and the Myxo just spread out until they find the E. coli, and then it goes into the E. coli because they start moving. They have other strategies to like spread out through the E. coli, It's actually another
Okay.
really cool property of the Myxo like collective behavior that they do when they're preying on other bacteria, which is they form these ripples. So it looks like the surface of water and it's like a wave across the surface, and then it's a standing wave, so it like oscillates. And it's very unclear of why or how they do this. It's another project that one of the grad students in our lab is working on figuring that out, but it looks really cool and it's a mystery as to why they do that. But
So I can get this wonderful video on YouTube, for example.
yes, there's there's a great video on YouTube that a lot of people show of like everything that Myxo does.
And do the E. coli run away? Do they sense it and flee? Or they just sit there and wait their fate?
So on the surface, E. coli can't move. They swim in media. I don't know what happened. I think Myxo can't swim. So
Okay, so if they're in 3 dimensions, the E. coli could go swim away. And
potentially, I don't know if they would detect it and swim away. I don't know if anyone's even looked at that. But
and Myxo, I'm sorry with all these questions. I know it's not the main part of your talk, but
Oh,
we
it's
also have only a soft deadline at 2 o'clock. So if you need to go over, that's fine. But does the Myxo grow up in a nutrient broth as isolated cells, or is always on a surface when it's
yeah, so with our
cultured?
experiments, yeah, we grow it. Actually, we first grow it on a surface, which so we have like these gel pads that they grow on, and then they will survive and grow on this pad for like a week. And then we take it from the pad and put it into liquid and let it grow in the liquid overnight.
Okay.
And do the experiment. So then we concentrate it from the liquid and put it back onto a gel of either high or low nutrients or whatever you're trying to test.
Okay.
And then they start depending on like the density and everything. So we've figured out for the future. Actually, so I'll just go to the next slide because
Sure.
yeah, so I'm studying them in high nutrients and then high density. So they get a monolayer that looks like this, or mostly monolayer. You have 1 or 2 layers.
Actually, Katie, I have 2 questions. Sorry, sort of following up on some of the things that David asked. So Myxo also go back and forth, right?
Yes, they do have a reversal.
What's the reversal frequency roughly in the situations that you've been looking at?
It's about 8 minutes.
8 minutes, okay. And they also leave a slime trail and that others follow on, right?
Yep, they do. We've started to look at those also a little bit because we can measure the 3D shape of the surface of our samples, which I'll show you more of later. But from our measurements, it looks like the slime trails are not actually, they do have like lipids and proteins and stuff left in them, but they're also a physical deformation in the surface of the gel. So they like dig out a track. So we kind of think that they're following the track. There's also a wetted layer, so there's water everywhere on this. So you have surface tension from the water coating everything. So like from a physics standpoint, it seems like it makes more sense for the water tension to be pulling them into these tracks.
The cavity, okay. But
they're digging behind, but we haven't like figured that all yet out.
I see. So that won't play a role in I'm guessing in what you're going to tell today, but maybe it will could potentially affect collective motility.
Yeah, it could. I think this time scale, so these experiments are done over about an hour. And you have cells covering basically everything. So I don't think the slime trails like do too much.
Okay.
It seems like they take longer to form. Like when you have a single cell digging out a track, they actually move a lot slower when they're trying to create a track through the gel.
I see.
So it takes kind of longer than this, I think, to form, to like start interacting with the tracks and like the whatever gunk they might be leaving in the tracks, I think.
Okay, thank you.
So we haven't really thought about the slime like in this context, but it's definitely there, yeah.
So just I'll share a very short follow-up from me. As you said, to Suraj, they can reverse direction. Are they polar? Do they predominantly go forward, or they go into reverse and equally likely to be going forward and reverse, and they just select the polar direction locally in the pack, or how does that go?
So that kind of depends on who you ask. I think for these experiments, like the reversals don't really matter too much. They do reverse every 8 minutes-ish in these conditions. I'll show you a bit in a minute, but we are trying to actually track the cells inside of the monolayers and be able to measure that.
Okay.
There are mutants that don't reverse, and they, at least to my eye, I haven't done a lot of analysis of the non-reversing mutants, but by eye, they look pretty similar to this. Like this is wild type, which does reverse.
George, our custom in these seminars is to, in fact, interrupt frequently, and if Peggy doesn't mind, I think we'll continue to do that. We also have a very soft boundary at 2,
Yeah.
so she can go on as long.
I don't have like too overly a lot of material, so the interruptions are good. I'm not worried about like time or anything. So
Okay,
I
thank you.
appreciate the questions. They're good. So, yes. Okay, so this is the experimental kind of what it looks like in the experiments that we did to look at this. So what we wanted to ask is how these cells start to form the second layer. So how do you go from getting one layer where your like surface tension is holding everything to your surface, and then the bacteria still somehow form a second layer? So we wanted to ask like, how are they forming the second layer? Oh, sorry. So, and we also want to learn about active materials from the context of like our experimental system. So we wanted to try to track, which is actually the next thing I'm going to talk about. So we're going to use some deep neural nets to segment all of the cells within the monolayer to try to track them. So we have these images of the cells, and then I, by hand, just spent a long time drawing a mask. So this is like my ground truth of where all of the actual cells are located. So this is a mask. The green is cells, and then it turns out to do better if you also label a border around the edge of the cells. That helps the machine learning kind of separate the ends of the cells when two are close to each other, and just helps in general. And then we have a background. So we have these masks. So we drew 23 images where we took the cell image and then drew the ground truth of where the cells are by hand on them. Then we feed these pairs of images into a U-net. So it's called a U-net because it looks like a U. But basically you take your raw like image of your cells, and then you downsample it and apply some filters. And you do this a couple times in however many layers you want to use. We did 3 layers, so it looks like this. And then after that, you can upsample until you get back up to your original image or back up through all your layers, and you get some predicted masks. So then the U-net outputs like a prediction. It tells, I think there's cells here and borders here. And then you take your output and you compare it to your ground truth that you drew by hand to get some loss function. So then we did this to all of our pairs of images, basically, and then you can update your parameters for all of your downsampling and filters with some learning rate. So you change all of your parameters a little bit, do it again, and then see if your loss got better or worse. And then if it got better, you like keep changing it in the same direction. If it got worse, you like undo your changes and try something else. And then eventually, after doing this a bunch of times, I think we did like 400 or something epics. So you converge on sort of a U-net that does a good job of like taking your raw laser images or your image of your cells and then giving you a prediction of where your cells are located. And this U-net and all of this machine learning was set up to work for our systems by Matt Black, who's a grad student in our lab. So after doing all this, we can feed our like videos and our whole images into the U-net and get a prediction of where our cells are, which looks something like this. So you can see by eye, it looks like it's doing an amazing job. You can see the red is where the cells are. So you have like nice long cells. It seems like it's segmenting them all perfectly. You can see the green borders. This is a probability mask, so that's why it's not all 1 or 0, but it seems like it's doing a really good job of segmenting the cells, and it is doing like a pretty darn good job, but so then we do some simple overlap tracking. So just look for overlapping pixels between frames of the cells, and you can track them through video. Hopefully it plays okay, but you can see most of the cells are being tracked pretty well across the time of this video, which is kind of short right now, but there is some flickering where the cells are lose, like merging and then dividing it, separating again. So we kind of lose IDs. So we can't track them all perfectly yet, but I think we're getting somewhere. And this was kind of like the first attempt, and it's already pretty good. So this is like what we're working on right now. So this is preliminary. But once we have all the cells being tracked well, we can do things like track clusters of cells and look at how their shapes are changing to see like what forces like groups of cells are applying, and connect the motion of individual cells to the more mean field treatment that we're doing of the system. So this is kind of a work in progress, but I wanted to like show you a hint of like where we're going to connect like behaviors of individual cells to like the material properties.
So Katie, can the neural net detect the presence of a second layer?
So I have changed up to do this tracking. We've changed the experimental setup a little bit to try to make sure that it doesn't form a second layer, because like you can see on this video, like around the edges of the second layers, you lose the cells, which kind of makes sense. And it also gets a little weird, like inside of the second layer, where like maybe the bottom layer and the top layer aren't perfectly aligned with each other, and you get like this crosshatch pattern. So we're trying to just look for this part of the project, we're trying to just look at only monolayers, because that's where you can make out the cells better. So yeah, it doesn't do a great job with second layers, but we can. So we basically use a slightly different media that has a higher surface tension, so it keeps the cells from forming a second layer as often.
Thank you.
Yep. So so that's kind of where we're going, but we don't actually have to be able to track all of the cells to be able to learn a lot about our system. Because we can use some concepts and like methods developed in liquid crystal physics. Liquid crystals are materials that are made out of rod-shaped molecules, and they have different phases that go from anywhere from crystalline, where all of your positions and orientations of your molecules are organized or well structured, to an isotropic phase where all of your positions and directions are completely random. Our system falls into this nematic phase where all of the molecule directions are aligned, but their positions are random. And in liquid crystal physics, you can't actually image all of your individual molecules in a liquid crystal. Instead, if you want to like study these materials, you do something, you look at data that looks kind of like this, where you're imaging a liquid crystal through a polarizer, and you can see the liquid crystals polarize light. So you can see like the average directions of the molecules. So bright would be aligned with your polarizer and dark is misaligned with your polarizer. So they study a lot about these systems by looking at kind of the average direction of your molecules. And in fact, if you take your molecules in your liquid crystal and you make them alive, so they have some active forces around them, which generally come in the form of a stretching force along the directions of your molecules, you get an active nematic. So this is a system of microtubules with molecular motors that walk along the microtubules and make them glide past each other. This is a video from the Dojig lab where they kind of first spurred this study of active nematics. And if you have, you know that your like active forces are stretching or compressing along your director field lines or the directions of your molecules, you can solve for all sorts of interesting flow fields and stuff within your molecule or within your material. And you can show that also these sorts of flow fields might be related to actual biological systems. So this is a system of neural progenitor cells where they showed that there's some points where cells tend to aggregate and some where they tend to deplete. So we're going to try to use this kind of framework to study our Myxo, and you can see that Myxo fits well into this framework because we have our rod-shaped cells. We can easily draw lines to see what average direction they're pointing in. So we're going to image the Myxo using this. Now I'll tell you how we're actually imaging these. So we have a 3D confocal profilometer that uses reflected laser light. So basically what it does is it takes a laser, shines it at the surface of your sample, scans an XY, and then does a Z stack and measures the amount of reflected light. And then by looking at each pixel, what height your reflected laser light is the brightest, you can find the height of your sample. So that gives us a height map that looks like this thing on the left. So you can see there's these holes where the height is 0. And then we have a first layer of cells where the height is approximately the width of a cell, and then a second layer of cells with a nice boundary, which is about 2 layers thick. So we can take this kind of height map and extract the number of layers at each point in space. And then at the same time, if you look at the brightness of the reflected laser light at each point, you can get a reflectance map of your surface of your sample, which looks like this, which is the videos I've been showing you. So by looking at this reflectance map, you can see the organization of all the cells within the layers. So we can simultaneously measure the height, which gives us a layer count of the cells, and then also a map of like the organization of the cells within these layers. And I'll play this video. You can see them moving past each other. So we can measure the heights and how these layers are forming, and then also how the cells are organized during this process. Okay, so we're going to take our reflectance maps to look at the organization of our cells, and using this kind of idea where we want to know the average direction that our cells are pointing at each point. We do this using the Hessian of our brightness image. So this is basically the lower eigenvalue, the vector associated with that gives you the direction of the lowest change in reflectance. So we have basically the direction that the brightness is changing the least in, which is along the cell bodies. You can see our color map here. So like this bluish green color is horizontal, like 0 or pi. And you can see that that falls on the cells that are horizontal. And then our vertical cells show up as this kind of pinkish orange color. So we can map out the direction, the average direction of our cells at each point in space using this method to get a director field. And then we can use the director field to calculate an order parameter, which is over some area that we picked, which tells us kind of the average or how much your molecules in that region deviate from the average direction. So this is going to be a value of 0 if all of your molecules are randomly oriented, or a value of 1 if everything is perfectly aligned. So you can see that there's mostly everything is aligned and you vary smoothly from one direction to another. But you have these points, which are the defect points, which I'll show you more in a second. But that's where all of your direction is undefined and you get all the colors meeting. So you have these defect points in the layer, and you can see that they match up with an order of 0, so low order where the defects are located. And I'll play this, hopefully it looks good on the video, but you can see how these defect points are moving around and our director field is changing sort of smoothly and slowly with time. And the defects are moving around, they form and annihilate in pairs. And so, yeah, so we can use this to locate all of our defects systematically within our cell layers and learn something about how the organization of our cells is evolving.
Okay, in that lovely movie that you just showed, how is theta ij bar defined? Is it some local thing or is it the whole global average of these?
It's local. So I just basically drew boxes on here.
Okay.
I forget how big exactly I picked the boxes. We had like basically picked a couple of maybe 5 by 5 or something pixels and then averaged the theta within that box and then compared like each pixel to that average direction.
Yeah, I guess you're going to shortly tell us which are the plus a halves and which are the minus a half.
Yeah, so so you can also measure the correlation length of the director field, which turns out to be about 16 microns, but just side note, I guess. Yeah, so the defects there's 2 types of defects. One of them looks like this kind of comet shape, while the other one looks like a nice little triangle. And yeah, so you can distinguish between them systematically by calculating what's called their topological charge. So this is the integral of the direction of your molecules as you trace a loop around the center of your defect. So for example, if you take this comet shaped defect on the left and you integrate your changing direction along that loop, here we have cells pointing horizontally, and then as you trace a circle around counterclockwise or positive theta, your molecules are also rotating with positive theta. And then when you get to the other side, you've done half a rotation. So if you divide that by 2 pi, you get a charge of positive 1/2. Whereas for the triangle defects, you might notice that as you trace this clockwise or positive theta, you actually have to rotate your direction the other way. So this ends up being a negative 1/2 charge defect. So we did this for all of our defects that we found in the samples and averaged together their director field. So this is experimental average of all of your director fields for all the positive defects, like aligned with each other and then averaged. So this is kind of our average shape of a positive defect, and here's our average negative defect. So you can see the nice little comet shape. So we're going to draw it as like a red dot with a line along its tail, and then we have our triangular shaped negative defects in blue. So now I will show you something about how these defects interact with each other. So this plot kind of shows everything that you might ever want to know about pairs of defects. So in the center of this plot on the left, for example, we have a positive defect, and then each point represents another positive defect. So this is for every pair of positive defects, we have a point. The color is the angle with respect to the radial vector, which is a little confusing. I'll show you a picture later of how these look, but the point is that you can sort of see positive defects have this region where there's no defects, so they repel each other. This dashed line is 10 microns, just that's kind of a guide. But this other plot is, there's a negative defect in the center here, and then we have each point is a positive defect near that negative defect. And then the color is the angle. So you can see that there's a very clear pattern, like they're orienting themselves very intentionally some way around the positive or the negative defects. The positive defects are, oh, it's confusing, sorry. Each positive defect is positioning itself in a very particular angle with respect to the negative defect when you have a pair of positive negative defects. This little inset is a density. So most of your positive defects sit along the triangular arms of your negative defects. You can look at the same thing for the negative defects. So this is where negative defects tend to go, around a positive defect or a negative defect. Again, you have this depletion, so they repel like charges. And then you can see the pattern in the colors again. So they intentionally point in some direction. And we have this high density along the tail. So you can measure the interaction potential with distance by calculating G of R, which is the probability distribution of each distance, and then normalized by like the radius or like the area that thing has to cover. So basically this shows that the red line here is positive pairs of defects. So positive defects interacting with other positive defects, and this high negative slope at short distances means that they're repelling each other. So you have a strong repulsive interaction between like charged positive pairs of defects and like charged negative, which is in blue. The purple is positive negative pairs. So pairs of positive interacting with negative defects. And you can see that in this case, we have a positive slope here. So that's an attractive interaction. So oppositely charged defects will attract. At short distances, it looks like there's a repulsive interaction because of this negative slope, but in fact, we just can't distinguish oppositely charged defects close to each other because they tend to annihilate very quickly when they get close to each other. So oppositely charged defects will annihilate. So short distances are a little questionable. Well, basically you have an attractive interaction between oppositely charged defects and a repulsive interaction between like charges, which is also what you see in passive liquid crystals. And then this interaction range, I kind of just picked by eye to be around 10 microns, which is, yeah, where the circle is drawn. So then this
Sorry,
is what,
Katie, can I ask another question?
yeah.
Is there a logarithmic repulsion, or your data's not good enough to say?
It's probably not good enough to say. Yeah, I'm not sure. Yeah, this is also something we're trying to work on and write up right now. So it's not, the analysis is still not completely done, but yeah, I mean, this is what it looks like. So not sure. So I'll show you now like what this angle actually looks like. So this is just a little cartoon of where each of the defects are, or sorry, if you have a positive defect at this location with respect to a negative defect, it will be aligned to this direction. So yeah, so you can see that like they're straight, like parts tend to point towards each other. And then if you have a negative defect around a positive defect, this is just the average angle that I showed you, like the color. This is what that looks like. Instead of as a color, it's as a plot of the defects. And now I'll overlay, this is what an actual negative defect looks like. So you can see there's streamlines, and you can see that like, it kind of makes sense looking at this plot as to why they would orient themselves in this direction, because like in the region between the two defects, this is like the direction, the director field lines kind of align between the pairs of defects. If that makes sense, hopefully. So yeah, so they tend to point towards each other, and the most common configuration, it turns out, is like this. So you have a negative defect with its triangle arm pointing at the tail of a positive defect. So I think this happens most often. This I'm also working on. This is preliminary, but basically you have a passive attraction between oppositely charged defects. And then you also have your positive defects are propelling themselves in active nematics, which has been shown in a lot of systems before. So your propulsion towards your head of your defect is opposing the positive or the passive attraction between them. So your defects kind of get stuck like this. So you have most of your defect pairs exist in this configuration because that's where like these are kind of pinned, if that makes sense. Whereas if you are in the other, configuration, this defect will propel itself and they'll just annihilate.
So, Katie, what's the mean separation between defects? Is that, can you remind me, the correlation length of your nematic was
Around 16.
I see. Okay.
So
And
yeah, I guess maybe that's a better interaction range, but yeah, this kind of approaches one towards somewhere between 10 and 15, but yeah, it is pretty noisy, this data, so hard to say exactly.
Because one would imagine that, you know, if the plus and the minus half defect are very close to each other, then the elastic attraction will dominate, and then this attraction potential will be negative, so they'll attract, and that's what will drive annihilation. But if if the plus half is run away and is self-propelling, and then eventually at further distances, one would imagine that it doesn't feel the elastic interaction as much simply because it's further away, and then activity should dominate and cause it to either decorrelate or effectively seem like repelling away from the minus half defect. But is that just, so is it just that the statistics isn't good enough that it all averages around around zero, so you don't see that or in this plot, or how should I?
That is definitely something we're trying to figure out right now. So I think that's right. Though I think maybe the theory is that the active propulsion of the positive defects is larger than the elastic attraction between like pairs or opposite pairs of defects at all length scales. So I'm not sure. I want to measure like the speed of the positive defects as they're moving, depending on whether they're pointing towards or away from a negative defect, because this is a little bit confused by it, because this does include the fact that the positive defects are propelling themselves. This is not just the passive attraction, right?
Yes.
Yeah, exactly. So we're trying to figure out how to like measure that more, but I think we can maybe start to get at it if we kind of break down this interaction potential to be like to include information about which direction the positive defects are pointing in at each distance. Yeah, so that's something I'm working on. I don't have that analysis done yet though, but yeah, good question. I think working on it. But yeah, Cool. So now we're gonna go back to looking at this layer formation again. So now we have all of our defects within our layers, and then they're moving around. You also have the layers and the holes opening and closing. So we can try to like look for a connection between the defects and the layering. So we spent a long time like staring at these movies, trying to tell what's going on, and started to notice this kind of thing happening, which I'll highlight. So here on the left is a positive defect. You can see it's comet shape in the cells, and I'll play the movie and you can see a new layer forms right at the center of that positive defect. And then here on the right, we have a negative defect, which moves over a little bit, and then a hole opens up at the center of the negative defect. So we saw this happening a bunch, and we were like, I wonder if there's something going on here, right? So we can look systematically at all of the events of a new layer forming, and then measure the distance between that new layer formation event and all of the defects of each charge. So the red points on here are that probability distribution function of the distances between positive defects and new layers. And then we normalize by some random uniform distribution of points to get like an enhancement of how much more often you're getting new layers close to positive defects than if it were happening by random chance. So random chance would be a value of 1, which is this black dashed line. And in fact, what you see in the data is that the positive, or the layers are forming close to positive defects about 100 times more often than you would expect them to if it were just happening randomly. And then it drops off pretty quickly with distance. And then we also looked at the distances between layers and negative defects, which are the blue points. And it happens randomly at long distances, but then we didn't have any data. So there were 0 events of a new layer forming within 10 microns-ish or 5 of a negative defect. So either it's suppressed and like can't happen at all, or it's just random if it happens, a new layer out of a negative defect, but we don't have the data to tell. So we have a very strong correlation between positive defects and new layers forming. And then likewise, if you look at the new hole formation events and the distance distribution to negative defects in blue and positive defects in red, you can see a similar relationship. So we have holes opening up at negative defects 100 times more often than if it were happening by random chance. Okay, so we've shown that now that there's a very strong connection between positive defects and layers and negative defects and holes. So we went to our friends in the theory department, and Ricard did all of this calculations and modeling for us to try to understand what forces might be occurring inside of the monolayer to drive this event or this process of layers forming at positive defects and holes at negative. So in this kind of active system, we have a force balance. So it's just very simple model where we have an active force, which shows up as a stretching along the director field lines, and it comes from the cells pushing against each other. So it's a stretching along the director field lines with a compression perpendicular to it, but that's our active forces from our cells. And then this active force is balanced by a friction. So friction, we have our friction coefficient times our velocity. So the friction coefficient in this case is actually anisotropic. So we have a larger coefficient of friction perpendicular to the cell bodies than along the cell bodies, which basically means it's easier for cells to be pushed forwards and backwards than it is to push them sideways. And we think this is because of the way the focal adhesions form and glide along the cell bodies. So it's easier for them to glide along the cell body than perpendicular to it. So we have this anisotropic friction, and then we have an active force, and the coefficient of friction and the active force both come from the director field, which we have in the data. So our only unknown here is the velocity. So Ricard took this model and then solved for velocity fields around each type of defect. And this is what he got. So this is a positive defect. You can see it's comet shape in the director field lines. And then this is the flow field that he calculated around the positive defects using this simple model with extensile act or stretching active forces and then anisotropic friction. And you can see in this flow field some interesting stuff. So first, all the arrows tend to point to the left. And then in the tail of the comment or behind the center of the defect, you have a larger amount of flow coming in towards the center of the defect than you have leaving out the other side. So this would suggest a kind of accumulation of material at the center of the positive defects, which is actually where we see the layers forming. So this seems like it could explain why there's layers forming at positive defects. And then if you do the same thing for the negative defects, what you'll find is that you have 3 zones now, because it's threefold symmetric. You have 3 zones of high flow coming out of the center of the negative defects with lower flows coming in the other directions. So you have a depletion from the center of the negative defects overall, which could explain the fact that new holes are opening at negative defects.
So
we
Katie,
have-
can I just ask, in this nice theory, do the Frank constants that would dominate the physics in the passive systems appear somewhere? Where
Yeah,
would the
so
Frank
that-
constant appear in this equation, the previous slide?
Yeah, that comes into the calculation of the shape of the defects, basically.
Okay.
So yeah, going from the director field lines that you have into the flow field is actually not affected. Well, this is not affected by the elasticity or anything in the cell layer, but that does affect the shape of this defect. So we just kind of guessed and used something for this model, but yeah, it can change it. It basically makes these either more pointy or like kind of more curved.
I'm just wondering if you couldn't back out the 2 different Frank constants that are almost certainly not equal in these bacterial systems. There would be one for bend and one for splay. And did you assume those are equal in the simple first cut here, or?
I think Ricard assumed they were equal, but I would have to check. I'm not sure. But we should be able to cut, because we have the average shape of a positive defect from the experiments.
Right.
So I think we could back it out by like fitting the director field to the experimental defects. It's a little challenging because we don't have a good way to measure the order, the actual like order parameter. Because the way that we measured it to locate the defects depends heavily on how big of a box you choose to average over. So like bigger boxes will give you a longer like decay of your order from the center of the defects, which like in liquid crystal physics, it should not depend on that. It should have to do with the organization of the molecules. So we haven't really figured out how to like actually measure the order. So it's a little hard to fit the model defect to the experimental defects, but it seems like it should be possible.
Yeah, and certainly if you can go in that direction eventually, I think it would be quite intriguing. I think that K1 and K3 are expected to be quite different for very elongated objects, at least in the passive case.
Yeah, I think that is definitely something to try to look at. I think, yeah, with this segmenting and tracking of the individual cells, hopefully we can also try to get at that question, since we'll actually have our like molecules.
Right.
And all of the molecules, but it's also a little weird because the defect core size seems to be smaller than the size of an individual molecule, whereas passive liquid crystals, you have a zone where actually all of your molecules are like randomly oriented and you have multiple molecules in that zone, so you can like you have something to average over, whereas in our case we don't necessarily have that, since it seems like all the cells are like organized around the center of the defect even. So yeah, that's something we're thinking about. We just haven't figured out the best way to approach it, but any ideas would be appreciated. But yeah, cool. Yeah, so then we took these, since we have these flow fields that would seem to predict an accumulation at positive defects and a depletion at negative defects, we went back to the experiments to try to measure the flow fields around the defects to see if it matched. So we used optical flow techniques to measure the flow field everywhere, and then took all of our positive defects, realigned them with each other, and then averaged them together to get the average flow field around positive defects, and the same thing for negative. So these are our experimentally measured flow fields around each type of defect. And you can see the same kind of things that I showed you in the model. So we have a high amount of flow coming in towards from the right to the left, towards the center of the positive defects. And a lower flow coming out the other side. So we have, in fact, a like convergence of material at the center of the positive defects, which is associated with new layers forming. And then similarly with the negative defects, we have these 3 zones with the high flow coming out from the center and the lower flow coming in the other directions. So that shows in the experimental flow fields, we also see the same depletion, which could explain that why our holes are forming at negative defects. So this simple model with active forces balanced by anisotropic friction seems to just explain why we have, or how this bacteria forms new layers and new holes at certain locations. And just to kind of illustrate that more, this is the net inflow at the maximum radius. So we drew a circle around the positive defects, and this is the what the flow field looks like. So if you integrate around this loop, you can calculate the net inflow and see that in the theory and the experiment, it's pretty closely matched, where you have a net positive inflow towards the center of the positive defects, and then a negative inflow or an outflow around the negative defects.
So in the theory bar over here, what all are being, what are the things that are being compared to and matched and fit to the experiment? So what's determining, so how do you actually do the theory calculation in terms of determining the friction, the anisotropic?
Yeah, so
The friction, the activity. So how do you fit for the various things?
So yeah, so we did do a fit. So I think that's in actually a different slide. So we took the center line of each type of defect and we looked at the X component of the velocity along that center line.
Okay.
For both the experiments and then also the model, so that we could fit those together. And those fits look like this. So we have our negative defects, the model fit is in blue, and the actual experimental data for that X component of the velocity along the X center line in gray. So this is the experimental data and then the fit on top of it. And so you can see they're very closely fit for the negative defects, and then the positive defects, the fit is in red here, and then the experimental data is in black. So when we were doing these fits, we noticed that there's actually this backflow in the experiments where the flow, which would be predicted to always go to the left, actually adds a little bit of flow to the right, or positive flow in the experiments. And so we spent a while trying to figure out why there would be this backflow in the experiments that is not accounted for in the model anywhere. And we figured out that if you actually include this pressure gradient, so now instead of assuming that everything's completely compressible, which is what we have been doing, if you assume there's some rate of like cells moving up into the second layer, which depends on the pressure, then you also have a pressure gradient, which gives you a force like from high density to low density. You can include this in the model, and then when you do that, you get this yellow line as your fit. So it's not essential to explain the layers forming at positive defects and holes at negative defects, but it does like explain some parts of the experimental data that if you include this pressure gradient in your model. So yeah, so maybe that is more of an answer than you wanted, but here's we did fit yeah, the experiment to the model using the center line velocity.
I see. And so you do this with the center line velocity along X, and then use this fit prediction with the parameters that you have in the force balance, and then use that to predict the net outflow, and that was what you plotted up here.
Yep. Yeah, and then we did it. Yeah, we checked a bunch of radiuses and then just looked at the maximum one, which is what's drawn here.
I see. Okay. And if you now take the fit of the flow along the center line and ask how, for the same values, does the flow look in the orthogonal direction? So along Y, how does it fare? Do the fit parameters work in other directions?
I I'm not sure that Ricard ever did that. I don't know. I think it does fit. I mean, if you just look by eye at the, like, here's the fit flow field and then the experimental ones. So they fit relatively well, I think. There's like some differences, of course, between the two, but
Okay.
I think it's pretty good at least.
Okay.
But-
Thank you.
Oh, relatedly, can you go back to that slide where you showed the fits?
Yes.
So what do you think is going on in like the positive X part?
Yeah, that's a good question. I don't know. It is definitely different between the model and the experiments. It's probably just noise in the experiments far away, but at least like qualitatively, they're both negative. So it's less like conflicting than having positive versus negative in the other direction. But I don't know. I'm not sure why they deviate so far out past like 15 microns.
I mean, I would imagine that, the calculations for a single isolated defect, that should only work on scales where you don't have other defects, right? And
That's
15
fair, yeah.
microns was about the separation between defects. So that's about also where the theory deviates from experiments. So maybe it's not so surprising.
Yeah, that's a good point. That is
Yep.
a very good point. It could be that. Yep.
Can I
Then
have
you
one-
could- Yeah.
can I have one more question on the active force? So it's a little bit unrelated to what's discussed before. So you mentioned we can calculate the active force from the direction field. Did I understand correctly?
Yeah.
So should I understand the active force as like coming from the interaction between cells?
Yeah, we haven't fully been able to connect, like, that's something we are working on, is connecting the actual individual cell motion or interactions with this mean field force. But yeah, generally it's comes from the stress in the material. So the stress is related to the like bend and splay in the director field. And then you can get the active force by just, taking the derivative of that active stress. And then we have, this is one of the fit parameters. So this is like our activity parameter.
I see, cool. I guess I just wish to ask, do we know kind of what's actually the interaction between cells in this animal, like how two cells communicate, or like is there a direct interaction between neighboring cells?
Good question. They do have some amount of cell signaling, so they can send like chemical signals between them. We don't think that is very relevant to like this process, because that is probably slower than the effects that we're seeing, or at least it's not necessary for the effects that we've seen just based on this model. But most of the interactions are just from physically running into each other and like pushing on each other. But yeah, they're also like, when the cells happen to, say they reverse and then they start moving away from each other, you get like a little gap, and then cells will come in from like neighboring lanes into those gaps. So I have a pet theory that is related to like how you get an extensile force from how these cells are moving. It's a good question. Like I am trying to figure out, like, this is definitely research direction I want to go, which is figuring out like how, because they are polar molecules, right? You have a cell that's propelling itself in one direction, but then somehow you're getting a mean field extensile force, so you have an active force in opposite directions, or like stretching like symmetrically. And how those two things connect to each other. That's part of why I'm trying to track all the individual cells, is to figure that out. So it's a good question, and I don't have an answer yet, but hopefully someday I will. So it is clear that when they interact with each other, they don't seem to care whether they're going the same direction or opposite directions. So if they run into each other, they're just as happy to like pass each other as they are to like flip around and go the same direction. So it seems like their interactions at least they're aligning, but nematically aligning. So they are head tail symmetric, the interactions, rather than a polar interaction where they want to go the same direction.
So Katie, is there any hope of using the genetics of the Myxococcus to knock to knock out genes and mess with some of the parameters in the simple theory?
Yeah, definitely. So we already are starting to look at cells that don't reverse. I don't have the analysis for it, but they definitely exist. We also have a few other things. You can also introduce drugs. That's another thing I wanted to start doing, which is like changing how fast the cells move by using like different drugs. You can also change stuff about the surface that they're on. But yeah, there's tons of things you can mess with, which is part of why like this is such a cool system to study these in.
So you said they reverse every 7 minutes on average, but if the dwell time going forward and backwards is similar, or is it more biased towards going?
It's the same. So when they reverse, they
Okay.
completely disassemble all the machinery at one end and put it at the other end.
Wow.
So they don't have like an actual head or tail, like overall.
Okay.
They just like completely reverse everything when they reverse.
Wow.
So actually, Katie, about your point about they because they reverse, they're not really propelling themselves in a single direction on timescales greater than 7 to 8 minutes.
Mhm.
So there are simple models of if you have a particle that just shuffles along its long axis more frequently than in the orthogonal direction, that you can compute a stress which looks exactly like the active stress that's written over here.
Oh, okay. Yeah.
So I think it's an interesting question now to ask, if you add a drug or you use these non-tumbling, non-reversing variants, and how will that affect either upregulating or downregulating effectively this, the activity over here, and either slowing down the dynamics or speeding it up and then getting something else.
Yeah, so definitely something that we're trying to look at now. We have done some experiments with the non-reversing mutants, so I can tell you kind of qualitatively what it looks like by eye, though I don't have the analysis done, but it seems like the non-reversing mutants have higher active forces, so the defects move around more and you have more pairs of or more defects. Probably, it looks like. So yeah, I mean, I'm not entirely sure what that means for it. It also seems like over very long times, like 8-10 hours. So these are over 1 hour what we've been looking at, but it seems like it eventually goes to a more polar state. So you get big streams of cells going the same direction. So I'm not sure like when or how that sort of transition takes place, but there are definitely differences between the reversing and the non-reversing, but you do see like half charge defects in the non-reversing mutant that like propel themselves and like at least qualitatively look very similar, though perhaps with a higher active force. But yeah, definitely something we're trying to look into.
Is there any hope of flipping the sign of zeta with drugs or genetics or something like that?
I would love to be able to do that. I don't know, like, yeah, I don't know. I think if you have like pushing rods that like move and push on each other, you will always get a positive like extensile system, but
Okay.
it is noisy. Like we don't think that the like active force is constant everywhere. Like we think that there's some distribution across the sample, which is like kind of complicated how we came to that conclusion, but basically another postdoc in the lab is studying traction forces in these cell layers. So he uses beads embedded in the gel and measures how they move. And he's found a lot of regions where you have a bunch of parallel cells and then the forces that they're generating are perpendicular to their bodies, which is very strange, but you could get something like that if you have a gradient of activity across like your cells. So we think that maybe there's a gradient of activity. So maybe with this like cell tracking, we can measure like more local active forces instead of assuming it's constant everywhere. So maybe locally you could have like negative or oppositely charged data or oppositely like contractile systems instead of extensile.
Thank you.
And just to remind myself that these experiments though with the multi-layering, this is not the starvation. This is not in starvation conditions. right?
Correct. This is high nutrients, yeah.
Okay. Do you have in the starvation response when you eventually form these fruiting bodies, is there a version of defect induced potential multi-layering which maybe centers into fruiting bodies? Is there any evidence, or I know Josh has been thinking about this for a long time.
Yeah.
I'm curious.
So sort of, this is, yeah. So we looked at high nutrients in this study because we wanted a lot of like layer formation events to study so we could get more statistics. If you're in starvation, they like form a layer and then another layer, another layer. You can't really image, like it's hard to tell what the cells are doing when you get past like 2 or 3 layers. We did have a couple of events where a third layer formed, and those all happened at positive defects also. So we haven't like been able to show anything along those lines, but yeah, I think that the positive defects like help localize where the layers form so that you can get more layers at the same point. So I think it is related. I think it is the same process when you go into the starvation. It just like maybe they pin themselves. I'm not sure. Like that is something to look at more, definitely. But
Thank you.
yeah.
Okay, so I just want to say, so it's around 2:15 now, so feel free to wrap up like maybe 10:15.
Yeah, I mean, I think I've actually shown everything. Like we can also look at the divergence of the flow fields and see, again, this is just another visualization of like the accumulation at the center of the positive defects. We have this negative divergence in both the experiments and the theory, and then positive divergence around the negative defects. But yeah, basically that was my summary was next. So yeah, so we have these cool system to study active pneumatic liquid crystals and myxobacteria, monolayers, which move around on a surface. And then we have the layer formation process, which is like crucial to the biology of the system, driven by the extensile active forces and anisotropic friction. Yeah, so I'll just put up some acknowledgements and I can
Thank you
keep
very much.
questions.
Beautiful.
Thanks.
Great, thank you for the wonderful talk. I know we had lots of questions, but are there any questions either from?
Do you know something about the dynamics of the second layer? So can they crawl on the first layer, or are they just direct along and point basically in the same direction as the first layer?
Good question. Oops, sorry. So we can't actually image like the different layers. We think we're just imaging the top layer. Here, I'll show you in this, maybe in this image you can see, I don't know, it's kind of small. Well, here, hang on, actually, there's a better one. Yeah, so for the most part, it looks like all the cells between the first and the second layer are aligned with each other, which I think is true because you can see small regions where there's a crosshatch pattern in the brightness image. And I think this is where the cells are misaligned between the layers. So it can happen where your cells are misaligned between the top and the bottom layer. But for the most part, it seems like they're all like in register. So they're all aligned with each other between the 2 layers, I think, but I'm getting this again from like just looking at this image. So I'm not sure.
But just to follow up on that interesting question, do the cells in the second layer independently reverse direction?
I have no idea. I don't know. I have no idea how I would even be able to, yeah, we can't like track the cells individually between different layers. So yeah, I can't say. I don't know. Yeah. Yeah, we're not even sure really exactly how like motion on the top layer compares to motion on the bottom layer. Maybe if we could track them, like, I don't know, we're not sure whether like the cell moving on top of another cell like moves double speed or something because they're both going in the same direction, or like somehow they're yeah, I don't know. We haven't, like, we don't have that kind of resolution, unfortunately, so it's hard to say.
in some systems, the bacteria, I think, can turn and then are perpendicular to the substrate. Do you see that as well, or are yours too long?
I think they're too long and they're too flexible. That does happen in other systems where it's like pressure driven, like from growth, I think, and then they pop up vertical. Yeah, but I think Myxo is just too long and skinny. We haven't ever seen anything like that. Maybe once they're in fruiting bodies, there's not a lot of imaging of the cells like inside of the middle of the fruiting bodies. So it's unclear how they're organized in there, but at least in like 2 to 3 layers, they're all flat and like parallel to the surface.
Great.
So, just check. Yes, sir. Any- That's all. Sasha, thank you. Yes,
Question.
go for it.
Question. Dimitrios.
Hi, sorry, wonderful talk. Quick question. So in your force balance equation, is this sort of like the force exerted on a single isolated defect? Or for example, is this stress that of a single isolated defect? Or is it this force, an average force from all, the bacteria around it in some way? I was a bit confused about that point.
This is an average force, so it comes from the director field. So it's like the mean field organization of the cells and the rate. Yeah.
Right, but the defects, they have their own sort of director field, right? Very unique
Yeah.
around them.
Yeah.
And they have a strong distortion on the liquid crystal.
Mhm.
So is there any other force other than that, or is it just the active force?
Yeah, so
I mean-
in order to understand the layer formation, at least, all you need is this friction and active force. They do
And
have
the other ones-
pili, Sorry.
Sorry, go on.
They have pili, which like are these appendages that they like reach out and grab and pull on stuff. So that is their other motility mechanism, which I guess I forgot to mention earlier. But we don't think that that's like very, like it's mostly dominated by the gliding, which is this active force. Yeah.
Okay, and using this force, you can also see the annihilation of defects, or not really? This is just for the formation, essentially, is what you're telling me.
So in the model, it's basically to calculate the flow fields around static like director fields. So this like doesn't involve any motion or like changing director field. It's just if you have a director field that looks like this, how is the material around them flowing? If that makes sense. So this doesn't actually have to do with the like formation of pairs of defects, really directly. Or
Okay.
it's just like,
And you meant,
yeah,
nice.
isolated.
So sorry, and you mentioned that you did some type of hydro calculation after that to see if this matches, the results of the usual passive. For example, for passive defects, what the flow field generated by an isolated defect looks like. So
Yeah.
you did that later. Okay. All right. Thank you. Sorry for the too
No,
many
you're
questions.
good. That's a good question. Hopefully I answered it. I'm not sure I understood. the
Yeah.
whole thing, but- Okay.
Yeah. Thank you.
Great. I guess, let's thank Katie again.
Thanks again, Katie. It was really lovely. Thanks
Thank
for your
you.
patience with the questions.
Good questions.
Be back in 2 weeks.